late. So welcome everybody to the dissertation, the doctoral dissertation defense of Bilgecan Sen. He's going to be presenting his work today, and I think you'll find it pretty exciting. This is just, I'm the chair of the committee, the defense committee, Stephen Baines. On the other people on the committee include Heather Lynch, and there she is waving, and Rajid Achkakaya was his advisor. And then joining us remotely is Arpat Orzgul, who's in Zurich. So he's a little bit further along in the day than we are. So what is going to happen here is that we're going to listen to Bilgecan present his work for about an hour. There'll be a public, an opportunity for the public to ask questions thereafter. At that point, the committee will refrain from asking questions. After the public has finished asking their questions, we'll log off and join a different link to have our private defense session where we'll ask our questions. Okay, is that clear? So with no further ado, I'm going to introduce Rajid Achkakaya. For some reason, I can't speak this morning. Achkakaya, who is going to introduce Bilgecan.
Thanks. It's a great pleasure to introduce Bilgecan for his defense talk. When Bilgecan first started with me, he had a lot of ideas about species distribution models and species interactions. As he was working on this, he also worked as an RA on a market capture data set that he'll tell you about in a minute. Before him, we had started analyzing these data, but he didn't like that we had this pragmatic but informal mixture of Bayesian and frequentist methods. I guess he's more of a purist, so he said, no, that won't do. So it must be redone with Bayesian methods. Now, we had tried that before, so I didn't think that it would work, but he made it work. And then he decided to do macroecology using the results. Now, I was never interested in macroecology that much because I thought it was more pattern than process. But Bilgecan proved me wrong again. He did that by demonstrating how these large-scale macroecological patterns emerge from local population level processes. He'll now tell you all about it. So Bilgecan, the floor is yours.
Thank you, Stephen. Thank you, Rashid. Thank you all for your kind words and welcome everyone. Thank you for being here. I am incredibly excited to be able to share this work that I've been working on, I think, in the past 6 years. I'm very excited to hear your comments and questions afterwards. Now, today's talk, as Rashid already told you, is about the relationship between small-scale ecological processes and macroecological patterns. Here I define macroecology simply as large-scale ecology. This largeness can come in, for example, in 3 dimensions, large in space, large in time, or large in number of species. And one of the most frequently recurring macroecological patterns is that species with large local abundances tend to occupy more sites. This pattern has a name. It is usually called the abundance-occupancy relationship. And in this hypothetical example of this relationship, you can see on the left the distribution of individuals of 2 species. Now, if we count these individuals on each cell, we can end up with these 2. We can create these 2 abundance maps. And then we can take the average abundance across all of the cells that a species occurs in. And then we can also count the number of cells a species occurs. And if we plot this against each other, these two parameters, these two variables, then we can see a positive relationship between abundance and occupancy like that. Now, this is also what we see with real data. On the left, you can see this pattern on British farmland and British woodland birds. On the right, you can see it on birds from US. This data on the right is coming from Breeding Bird Survey. And on both cases, you can notice the strong positive pattern. Now, there's been a lot of different processes suggested that that can explain this, that potentially can explain this relationship. And Beauregard and Rothbeck counted 13 of them in their review in 2010. For example, some researchers, some ecologists argued that this was all about metapopulation dynamics. This was all about dispersal. This is how dispersal is dominant in these processes. That's when you get this positive relationship. Or this is about sampling bias, some argued. It was all about how we measure abundance or how we measure occupancy. What I'm going to be interested in today and what I'm going to talk a lot about today is called vital rates mechanism. Vital rates mechanism is special because it's the only mechanism, it's the only process among these 13 of them that tries to explain this positive abundance occupancy relationship using species demography explicitly. Now, I will talk about vital rates mechanism in detail, but before I do that, I'd like to talk a bit about intrinsic growth rate, because I will also mention this concept throughout the presentation a lot as well. Now, intrinsic growth rate is sometimes considered as a species level density independent maximum population growth rate. And this is, for example, you can see how it is depicted in the typical logistic equation growth. This is a very typical formula that you can see in a population ecology textbook. However, it doesn't have to be this way. Intrinsic growth rate can also be modeled to vary temporally and spatially as a function of the environment. Now, in this definition, it is still density independent, and you can still think of it as a species level maximum in a way. It's just that this maximum now can vary across space and time depending on the environment. And this is the kind of intrinsic growth rate that Holt et al. In 1997 used to explain their proposed vital rates mechanism. Now here, intrinsic growth rate is simply birth rate minus death rates. Death rate can change across species, along species. I here signifies a given species. Birth rate, however, not only changes across species, but it can also change as a function of the environment. Here, X signifies that. X can be different among different populations. Now, as a result, intrinsic growth rate here is also can change across species, but also it's a function of the environment as well. Now, in vital rates mechanism, intrinsic growth rate works as an intermediary between abundance and occupancy. So in the first step, I'm going to show you how intrinsic growth rate is related to occupancy. In this very simple case that is inspired by Holt et al. 1997, we have on the y-axis the birth rate, and on the x-axis environmental gradient that is represented by the x here. And let's say that this birth rate increases to an environmental optimum, and then as you move farther from the environmental optimum, this birth rate decreases. Now, you could show this with a typical bell-shaped curve as well, but I'm just going to use this triangle shape here just for simplification of the relationship that I'm about to explain. Let's say that for our first species, there is this single death rate that does not change as a function of the environment. Now, just by looking at this death rate and birth rate, we can actually understand where the species can occur. Because species can only occur, a population can only occur when its birth rate is higher than death rate. So we can look at where these two curves actually intersect. And this place that they intersect gives us the range of the environmental gradients that our first species can actually exist. If we have a uniform and homogeneous environment, this range of environmental gradient will translate directly into occupancy as well. Now, let's say we have a second species with a higher death rate. Now, because this has a higher death rate, what's going to happen is that it's going to have a smaller range of environmental gradients that has higher birth rates than its death rate. So if we have, again, a homogeneous and uniform environment, we can say that in this relationship, the first species is going to have a larger occupancy than the second species. So we can say in this relationship, if this is true, higher intrinsic growth rate can lead to higher occupancy. Now, the second part, I'm going to relate intrinsic growth rate into abundance. In order to do that, Holt et al. Used a different formulation than logistic equation growth, the typical logistic equation growth. It's an alternative parameterization. Here, there is no carrying capacity. Instead, there is this parameter, density dependence parameter called mu. This mu is simply the per capita effect of abundance on intrinsic growth rate. So it tells you how much the intrinsic growth rate will increase if you just add a single individual to this population. One of the nice features of this equation is that it's equilibrium abundance. And in a way, you can think equilibrium abundance as carrying capacity. Its equilibrium abundance is actually determined by the intrinsic growth rates and density dependence strength. So if we have, again, like multiple species, and if those multiple species have similar density dependence strengths, then higher intrinsic growth rate will again be associated with higher equilibrium abundance. We have already established that higher intrinsic growth rate is related to higher occupancy. Now we can say that higher intrinsic growth rate is also related to higher abundance. And in this indirect way, higher occupancy is indirectly related to higher abundance, which creates that positive abundance occupancy relationship or expectation of it. Now, this is a very simplified example, and in their paper, Holt et al. Goes into great detail to explain how this mechanism can actually, or not the mechanism, but how the positive abundance occupancy relationship can break down depending on these depending on these demographic parameters. So they do talk about a lot of different scenarios, and I would like to summarize them further. So we have the abundance occupancy relationship. This could be a positive relationship, or it could be a really weak relationship, or simply we can say that there is no relationship. There are extreme cases where you can actually see a negative abundance occupancy relationship too, but I'm not going to be talking about those today. So if we have one of these two relationships, there are certain expectations or predictions coming from the vital rates mechanism. The first one is the correlation between abundance and intrinsic growth rate on the population level. Now, if there's a positive abundance occupancy relationship, our expectation is that this correlation should also be strongly positive. However, if the abundance occupancy relationship is weak, we also expect this correlation to be, maybe it's weakly positive, but more likely it's even there is no relation, there is no correlation between the two. The second one is that the density dependent strength across species. This strength, if we see a positive relationship, should be quite similar across species. If we see a weak or no relationship, then our expectation is that it should be different among species. There's also the response to the environment. If we see a positive relationship, our expectation is that for the group of species that we see this abundance occupancy relationship, this positive relationship, their response to the environment should be similar. For example, both of their vital rates should increase with increasing annual mean temperature. However, some species respond to the environment differently here. Then we expect this relationship to be more, this pattern to be more related to a weak or no relationship of abundance and occupancy. And then there is the distribution of the environment that I talked about. If there's a positive relationship, we expect to see a more homogeneous and uniform gradient of the environmental variables. If we see, for example, in the extreme cases like islands or mountaintops, then we don't necessarily expect to see a positive abundance and occupancy relationship. And the last one is the source sink dynamics. If there's a positive relationship, the vital rates, the prediction of the vital rates mechanism is that most of the populations here are doing fine by themselves. We can call them all sources. Yes, there are dispersal between them, but they can exist on their own. But if the sourcing dynamics are dominant to such degree that, for example, in the simplified case where we have a single big source population, and then a lot of the occurrences are actually determined by dispersal from the source population, then we also expect this to be related more to weak abundance-occupancy relationship. Now, this is a lot, and this is a lot to actually test in a single dissertation. It requires a lot of data, but also a lot of different kinds of data. So what I'm going to do today is to just focus on the first two that is explicitly related to species demography. In order to do that, like Rashid told you, I'm going to first introduce to you a new framework to estimate vital rates, abundance, and density dependence. In the end, these are the parameters, these are the demographic parameters that we need to be able to test the predictions of the vital rates mechanism. Now, in this chapter, I have also done extensive simulation studies to show that this framework works. However, in the interest of time, I'm not going to show you those results. So I'm just going to use the first chapter to set the stage. But if you're interested, we can talk about this after the presentation. Now, in the second chapter, I'm going to do just what I told you. I'm going to use this framework to test the predictions of the vital rates mechanism. And then in the third chapter, I'm going to talk about species distribution models, because I think it's a great sin if we don't talk about species distribution models while we're also talking about abundance and occupancy. In the end, this is what species distribution models is all about. It's about abundance and occupancy. And I'm going to look at how species distribution models measure up against species demography. And in these two chapters, my general message is going to be positive. Positive in the sense that, yes, we can have some sort of link between small-scale processes and more large-scale macroecological patterns. But on the last chapter, I'm going to shift gears a bit, and I'm going to talk about not spatial patterns, but I'm going to talk about temporal patterns. And in this last chapter, I'm going to tell you the limitations of this small-scale process-based approach. All right, let's begin with the framework. This framework is called CJS-pop. The CJS names comes from Cormack-Joly-Sieber. It's a mark recapture model, and Cormack-Joly-Sieber is the most basic, the most fundamental mark recapture model. POP is there to signify the purpose of parameterizing population models. So that's why we created CJS-pop in the first place. CJS-pop can estimate stage-based survival, fecundity, density dependence, and environmental stochasticity. And it can do that using only, using only mark recapture data. So one of the advantages of CJS-pop is that it's quite data efficient. And I'm going to repeat what Rashid said. They already had started this work when I arrived in the Akçakaya lab. So a lot of the things that I did in this chapter to build up on what was already built, already built on great work. So this is the basic structure, basic structure of CJS-pop. On the left, you can see the directed acyclic graph. So it shows you, this graph shows you how the data and the parameters are interrelated. Now, I use Bayesian statistics to calculate the parameters involved in this framework. And now once I have these parameters, we can use these parameters to actually build a population projection matrix. And the data that I use to apply to CJS-pop is coming from a program called Mapping Avian Productivity and Survivorship. This program is organized by the Institute for Bird Populations since 1989, and they're operating across 1,200 bird banding stations across the United States and Canada. The time period of the data that I've worked on for my dissertation comes from 1992 to 2008. I only use data from conterminous United States, so no Canada, no Alaska. And I define a MAPS location, which is just a collection of bird banding stations. I define a MAPS location as a natural population. So across this presentation, when I say a population, what I mean is a MAPS location. Now, because in the beginning, I told you that the intrinsic growth rate should be here should be determined as a function of the environment. This is how I also modeled survival and fecundity in CJS-pop as well. So they are not only modeled as functions of density, but also as functions of weather-related variables. Here are these 10 variables that I use to model, to use in CJS-pop. However, I did not want to add these 10 variables as they are to survival and fecundity. That's a lot of variables. So I did, for a given species, I did PCA with all populations in years, and I selected PCA dimensions that explain 80% of the variation, and that reduced the dimensionality a bit. So when you have a stage structure matrix and it has 2 stages, like adult and juveniles, you can calculate adult survival, juvenile survival, and fecundity, and you can use these to calculate the growth rate of a population. Because all of these demographic parameters are functions of density, estimated as functions of density with CJS-pop, I can set the density to 0, and in which case I actually can estimate their density independent values. And when I estimate their density independent values, then it means that I can actually estimate intrinsic growth rate. And that's what I do. I estimate the intrinsic growth rate of each population in each year for a given species. And I average this intrinsic growth rate in a population across or between the years of 1992 to 2008. And I call this R bar. And then I take the median across all populations, across all R bars, and I call this R double bar. This is a species level intrinsic growth rate. And I use median here just in case that there are some populations with really large R so that it doesn't affect my species level estimate too much. I do a similar method when I conduct a similar method when estimating population abundance. I use the heuristic estimator of market capture data. Here you can simply divide the number of captured individuals to capture probability, and capture probability is estimated in any market capture model, and of course it's estimated in CJS-pop too. Now in CJS-pop, however, capture probability is a function of capture effort. So not only we have varying numbers of captured individuals across years and populations, we also have varying capture effort. With that, I can estimate abundance of each population in each year. And similarly to intrinsic growth rate, I can average this time series of abundances between the years of 1992 and 2008. And I call this N bar. And I can take the median across all populations, across all N bars, and I call this N double bar. So these are the basic parameters, demographic parameters that I will be using throughout this dissertation. And I apply this CJS-pop method to 17 species. Now, these 17 species are generally common species. However, there's a good bit of variation in terms of their occupancy. And that is necessary because if they didn't vary in their occupancy, in their range, then it will be impossible to look for a relationship between abundance and occupancy, right? So they have enough variation for me to be able to explore abundance-occupancy relationship. Now, this is the framework, this is the nutshell of CJS-pop and how I use it. And like I told you in chapter 2, I am going to use CJS-pop to test the predictions of vital rates mechanism. I don't know yet, however, I didn't show you yet, if there's a positive relationship or weak or no relationship between abundance and occupancy for these 17 species. Depending on that result, our expectations are going to change. If I, for example, see a positive relationship between abundance and occupancy, these will be my expectations. If I see a weak or no relationship, these will be my expectations, right? So that is the first thing that I need to set. So here in these graphs, I am showing you on the x-axis geographic extent. I am going to use 2 different metrics of occupancy. The first one is geographic extent. I calculate geographic extent as the area of 100% minimum convex polygon of all maps locations. On the y-axis, you have N double bar and R double bar. These are all on log scale, including the geographic extent. Each point in these graphs is a single species. The orange line is the regression line, but it is only here to show you the general trend of the data. Now, I calculated the correlation coefficient between these two variables, among abundance and geographic extent, and intrinsic growth rate and geographic extent. And you can see this here, row, those estimates. And I estimated these in a Bayesian framework. So because this is a Bayesian framework, this estimate itself has a posterior distribution. And because it has a posterior distribution, I can actually assign a probability to our hypothesis. Our hypothesis, coming from the expected abundance of occupancy relationship, is that this correlation should be positive. And when this probability that rho is positive, is close to 1, then we can say, yes, we have good evidence that there is a positive relationship. Or when it is close to 0, we can say that we have almost no evidence. In fact, there is potential that this is a negative relationship. And what you can see here in these both cases is that there is almost no relationship between species level abundance and geographic extent, and between species level intrinsic growth rate and geographic extent. Now, we don't have, just looking at this, there's weak abundance occupancy relationship or no relationship. And because intrinsic growth rate is not really related to geographic extent, we can say that intrinsic growth rate, just by looking at the data, cannot really, or does not really play the role of intermediary for this data. Now, the second metric that I'm going to use for the occupancy is number of populations. So this is the number of MAPS locations that a species occurs in. The data here, the results here are a bit different. For species level abundance, and again, these are all on log scale, every point is a species, and the orange line is the regression line giving you the trend. And if you look at this relationship between abundance and number of populations, you can see that compared to the previous graph, rho is a bit higher. It is 0.26. And the probability of a positive association here between abundance and occupancy, probability of positive correlation, is also slightly higher, is 0.86. There is, however, a negative relationship this time between intrinsic growth rate and number of populations, but there is a very weakly negative relationship. And of course, the probability of there is a positive association is quite low. Now, in the abundance occupancy literature, however, these relationships between abundance and occupancy is usually reported to be really high. For example, in the first couple of slides that I showed you, that relationship, those relationship was actually upwards of 0.5. Some of them were 0.7 correlation. So the correlation strength was quite high. So seeing this correlation strength, and again, not really that high of a support and evidence for positive association, I think it is safe for me to conclude that the route that I should go here is that there is weak or no relationship between abundance and occupancy. So now I have specific expectations about the correlation between abundance and intrinsic growth rate and how density dependent strength varies across species. Now, if vital rates mechanism is true, then its predictions here should be consistent with the results that I'm going to report on these two different expectations, these two different predictions. So let's first start with the correlation between abundance and intrinsic growth rate. Here in this graph, it is different from the previous ones because I am showing correlation coefficients for each species separately. On the x-axis, there is the correlation strength between population level average abundance and population level average intrinsic growth rate. And on the y-axis, you can see the 4-letter code for each of these 17 species. In this graph, the orange dots are the point estimates of the correlation coefficient. The black line is the 50% credible interval, and the gray line is the 95% credible interval. If you just look at the orange dots, you can see that they are pretty much distributed all around the correlation coefficient strength of 0.25. And of course, there are exceptions, for example, such as granted. This is interesting because it is actually quite close to the correlation coefficient that I estimated between species level abundance and number of populations. So we can say, just by looking at this, there is really a weak positive correlation between population level average abundance and population level average intrinsic growth rate. But it's also that these correlation strength estimates are quite uncertain. Most of them actually intersects, overlaps with 0. So even in the cases that they are positive, we are not 100% sure that there is a positive relationship between population level abundance and population level intrinsic growth rate. So we can say that our first expectation is met. There is a weak correlation between abundance and intrinsic growth rate on the population level. And for some species, we can even say there is no such relationship at all. There is no correlation at all. Now, the second one I find a bit more important, density dependent strength across species. It is also a bit trickier to actually show, because in their paper, Holt et al. Did not actually quantify what they mean by similar density dependence or different density dependent strength across species. But there's still few lines of evidence that I can actually follow to see if the results that I produce are consistent with the predictions of the vital rates mechanism. Now, in this graph, let's first focus on the right-hand side in this graph. And the y-axis, there is the species level median intrinsic growth rate. And on the x-axis, there is the species level median abundance. Each point is here, again, just a single species. The orange line is the regression line, giving you the general trend. Rho is the correlation coefficient, and this gives you the probability that this is positive. This correlation strength is positive. Now, if there was a positive abundance occupancy relationship among these 17 species, our expectation for the species level demographic parameters, our expectation was that this should also be positive. But it is not. It is, in fact, strongly negative. And this negative relationship was predicted exactly as is by Holt et al. They said that this kind of relationship, if they are related, if this relationship is related to density dependence, then this is what might break down a positive abundance occupancy relationship. They said a species with small intrinsic growth rate but weak density dependence can obtain enormous local abundances compared to another species with large intrinsic growth rate but intense density dependence. So I'm not showing you yet about anything about density dependence, but just in terms of that trade-off like appearance, this is what they exactly predicted, and this is what we are seeing in this graph. So now let's look at what the density dependent structure looks like. Now, their prediction was related to how density dependence was related to, is related to abundance, and how density dependence is related to intrinsic growth rate. And here in these graphs, on the y-axis, I'm showing density dependence strength on survival. This is y-axis is on logit scale. And species level species level abundance and species level intrinsic growth rate are again on log scale. Each point is a species, and orange lines are giving you the regression lines, the trends. Now you can see that between the density dependent strength and abundance, there's a strong negative relationship, such that if you have a high abundance, generally high abundance, then that species tends to have a weak density dependent strength. Or if you have a high intrinsic growth rate, that species tends to have a higher density dependent strength on survival. Now, this is exactly as what was predicted by Holt et al. So this is not just a matter of, are these density dependent strengths different enough? This is also a matter of, they are different in such a way that they are actually able to produce the pattern that was predicted by Holt et al. In 1997, that was predicted to break down the abundance occupancy relationship, the positive abundance occupancy relationship. Now, I find this exciting and fascinating because when Holt et al. Explained this, when proposed this in 1997, they did not use any data. They did not use simulated data. They did not use real data. It was just a theoretical concept. And they used a completely separate mathematical structure than CJS-pop. And here I am in 2020, I'm using state-of-the-art market capture models to show and a model that is completely different from from what Holt et al. Used to show that the results that I obtained is actually quite consistent with what they predicted. I really find it exciting. Of course, this is ecology and nothing is that straightforward. So now we have this density dependent strength coming from fecundity. Now on the y-axis, I'm showing the density dependent strength on fecundity. This is also on log scale. So what you're gonna notice, there is exactly the opposing patterns in survival and fecundity. So for survival, the predictions of the vital rates mechanism of Holt et al. Exactly holds, but for fecundity, it does not. However, the relationship between the density dependent strength and these demographic parameters is stronger for survival rather than fecundity. So I suspect that the density dependent strength on survival is actually the leading factor in giving us the pattern between the negative pattern between species level intrinsic growth rate and species level abundance. So this is still in general, I believe, good news. I would like to also add that this is the first time that I see that such opposing pattern in density dependent strength in survival and in fecundity. And this is definitely going to be one of my future obsessions to understand why in some species, in these species, that we see such opposing patterns. So in the end, I can say that using CJS-pop, I actually showed that these two predictions, these two expectations coming from the vital rates mechanism, if we see a weak abundance occupancy relationship, is consistent with the results that I obtained. Now, this doesn't mean I have conclusive evidence that the vital rates mechanism is the mechanism, is the process that can explain all abundance occupancy relationships. Of course, there are still other processes that I need to test. However, I think this is a good first step in the right direction. So in terms of interim conclusions, I can say that the results are consistent with the predictions of the vital rates mechanism. But most importantly, I can say that demographic processes for macroecological patterns can be disentangled. We don't have to do just hand waving and say large scale patterns, are established by incredible interactions between a lot of parameters and we cannot disentangle them. So macroecology is all about studying patterns. This is true in some ways. However, I think it is good to have specific, simple, and testable hypotheses about small scale patterns. We can still learn from these. And I believe I showed you a simple example of that. And of course, this doesn't mean that it's all about small scale processes and it's all about isolated hypotheses. Of course, I would like future studies to explore more about how these different interactions between demographic processes actually give rise to, for example, different macroecological patterns, such as abundance occupancy patterns. Now, one of the things that I'm really interested in is to look at how dispersal actually interacts with density dependence, strength, density dependence on vital rates, something that I hope I will be able to do in the future. Now, in chapter 3, like I told you, I want to talk about species distribution models because they are inherently related to abundance and occupancy. In a typical species distribution model, you have your occupancy data, usually as presence and absence. You relate that occupancy data to some environmental variables using some form of statistical modeling. And as a result of those models, you make spatial predictions. So you start with the occupancy, you start with the distribution of a species, and then at the end, you make predictions about the distributions and the occupancy of that species. However, usually people who do species distribution models, they don't just see this as a phenomenological model. It is a phenomenological model, but they also think about this as fitting the niche or quantifying the niche. And this is interesting because even though species distribution models is a phenomenological model, the niche concept is quite mechanistic. It is quite process-based. It's all about survival, reproduction, and resource use. So there is a relationship, there must be a relationship, if this is what we're doing then, between species distribution models and species demography. This is not only about occurrence. And of course, I'm not the first person to make that observation. And one general idea and I'm gonna spare you the theoretical details, is that species distribution models predict species abundance. So whatever the output of a species distribution model is, you can call it probability of occurrence or you can call it suitability. If it's that higher, then it also means it's predicting higher abundance. Now, unfortunately, the evidence on that is quite inconclusive. On the left, I'm showing you results from a meta-analysis study across 450 species. The correlation coefficient here is between an SDM output and abundance. And you can see that the correlation strength here, again, tends to be weak. It is below 0.5 in general for most of these species. Also, and in even a worse scenario here, in a more recent paper for mammals and plant species, Dallas and Hastings showed that there is really no discernible relationship between abundance and SDM output. Here for mammals, it could be really negative, it could be really positive, but mostly it's just centered on zero. So the plant species, there is not even those strong relationships, it's just centered on zero. So the evidence is really inconclusive here. And for intrinsic growth rate, of course, the second demographic parameter that researchers, ecologists try to associate with species distribution models, the results are pretty much the same. Now, we know that intrinsic growth rate is related to species occupancy. It's all about source and sink dynamics. So SDMs can also be related to intrinsic growth rate. However, in this study in 2014, you can see if you look at these distributions between, again, the correlation coefficient between intrinsic growth rate and the output of a species distribution model, they are pretty much centered on zero. There is some tendency for intrinsic growth rate to have a negative relationship between with the probability of occurrence, but that relationship is still quite weak. Interestingly, however, this is the only study that I was able to find that compares SDM outputs to intrinsic growth rate. There is no such study on animal species. So the results that I'm going to present you today is the first time that there is a comparison between intrinsic growth rate and species distribution models for animal species. So in order to do that, I've used two different species distribution models. One of them is hierarchical modeling of species communities. This is basically just a Bayesian GLM. And I used linear effects when building this model so that its structure was more similar to a CJS-pop. But I also used a more typical model, boosted regression trees. It's an algorithmic-based model, so it can fit nonlinear responses to environmental data. So these are the two variables that those are the two models that I used to fit species distribution models. And basically calculate the species distribution model outputs. So in order to explain to you like what I have here to make those comparisons, let's say I have this simple landscape. These orange squares are where I see the species. These are the populations of these of a given species. And for each of these populations that a species occurs in, I have some demographic parameters coming from estimated by CJS-pop. You're already familiar with the population level average abundance, population level intrinsic growth rate. In addition to that, I calculate the 95% quantile of the abundance time series of a given population. This represents the maximum value the maximum abundance a population can get. And I also look at how probability of intrinsic growth rate is higher than zero. Here, zero is important because since intrinsic growth rate is centered on, it's on log scale. If this is lower than zero, then it's a sink population. And if it's larger than zero, then it's a source population. So this gives you the probability of a population being a source. And then I have the probability of occurrence estimates coming from these two SDMs, HMSC and BRT. So in order to compare species distribution model output to demography, I do simple linear regression in a Bayesian framework. Here, demography is the response variable. I have the intercept alpha and I have the slope beta that associates probability of occurrence to demography. And because this is a Bayesian framework, I have a posterior distribution here for beta. And my hypothesis is that there should be a positive relationship between demography and species distribution model. So this is the probability, this is the hypothesis that I assign the probability to using the posterior distribution. This is the probability of positive association between demography and species distribution model outputs. This is how I'm going to determine if there's a positive relationship between the two. However, it's not only about a positive relationship, it's also about how much spatial variance of demography is explained by species distribution models. And here I am going to use a typical metric that is R squared. And as the last bit, I am going to compare the probability of occurrence values, the distribution of probability of occurrences between the source populations, between the populations that has intrinsic growth rate larger than 0 and sinks that has the intrinsic growth rate lower than 0. Okay, so here's the first bit of results. Now, in these graphs, in these plots, you can see on the x-axis, the demographic parameters that I just mentioned to you, the intrinsic growth rate, the probability of a population being a source, the average abundance or the population level average abundance, and the population level maximum abundance. So if we ask, is there a positive association between the two? For abundance, we can say that yes. In the y-axis, I am showing you this the distribution of the positive association, distribution of the probability of positive association. Each dot here is again is a single species, and box plots is giving you how these distributions look like. So if you look at these for abundance-related parameters, you can see that for both cases, for HMSC and for BRT, these probabilities of positive association are quite stacked up against 1. So there are a lot of species with good enough evidence that there is a general positive association between the two. This pattern is a bit stronger for species distribution for BRT, because there are those species that this probability is actually close to 0.6, that those probabilities are close to 0.6. Now, when we look at the intrinsic growth rate related parameters, however, yes, there are some species that definitely has some positive association between species distribution models and intrinsic growth rate related parameters. However, there are also some species that has definite negative relationship between the two. And these results are quite similar to what Tullier et al. Found in 2014. And I find that to be interesting because there are only 2 studies that I'm aware of that does this, and both find a tendency for intrinsic growth rate to have a negative association with the species distribution model outputs. This negative association, however, is a bit weaker for BRT if you compare this section of the plot of BRT to this section of the plot to HMSC. Compared to the association of the with the abundance-related parameters, you can still see that this is pretty much more negative. There are some species with pretty much a more negative relationship. Now, this determines how the general association between SDM outputs and these demographic parameters. However, it's also about how much variation these species distribution models actually explain in demography. And unfortunately, for me to see that species distribution models really do not explain that much variation in demography. Here, for example, if you look at, these are the same kind of box plots, by the way, and on the y-axis, you can see the R squared value, and each point is here, each dot is a single species, and this is showing you the distribution of R squared values across 17 species. If you look at the median values of those distributions, they are close to 0.1. So for a lot of the species, for both HMSC and for both BRT, there is a lot of unexplained variation here in species demography. So just using species distribution models to explain that variation, spatial variation in demography basically is just not enough. There are exceptions, however. For example, this outlier here is Carolina chickadee, and it has more than 0.5 R squared value, which is actually surprisingly high for me. I wasn't expecting to see that high values for any of these species. Unfortunately, however, this relationship for Carolina chickadee between the SDM output and intrinsic growth rate is actually negative. So even though species distribution models explain a lot of spatial variance in intrinsic growth rate for Carolina chickadee, the relationship is completely negative. And I think that's also an interesting result, but it's also a bit hard to explain. Now, the thing that I care most about from these results is the question that can SDMs distinguish sources from sinks? In this plot, on the y-axis, I'm showing you the distribution of the probability of occurrence for sink populations in gray and for source populations in orange. In the x-axis, you can see 11 species. I use not 17 species and 11 species for this plot because 6 species did not have any sink populations as estimated by CJS-pop. So this is going to be limited to these 11 species. So just in general, if you look at compare these distributions of probability of occurrence between sinks and sources, you can see that in general, HMSC is having quite a hard time distinguishing between the two. Now, it is doing a bit well for brown creeper. Oh, by the way, I forgot to mention the numbers that you see above is the p-values that comes from a simple t-test that compares the averages, the average probability of occurrence of a source population and the average probability of occurrence of a sinks population. So if you look at BRCR, the brown creeper, there's actually a significant difference between the two, which is at least we can see that for brown creeper, and that's good news. However, if you move to Carolina chickadee, well, we already know there's a negative relationship. So of course, this is also reflected in the fact that the CJS-pop actually has, sorry, the SDMs actually give, HMSC actually gives higher probability of occurrence values to sink populations compared to source populations. But in general, for HMSC, there is no, I think, general ability to be able to distinguish between sources from sinks. The results, however, get a bit better with boosted regression trees with BRT. So this is the same kind of graph. And for these 11 species, 7 of them actually have significant differences between sources and sinks. And those differences is in the correct direction. So BRT, on average, can give higher probability of occurrence values to source populations compared to sinks. Here, for example, we can see the same relationship with brown creeper. Sources have higher probability of occurrence than sinks. Here we can see that for Carolina chickadee, remember for HMSC, there was quite high probability of occurrence estimates. For sink populations, this is definitely not the case for BRT. And we can see that for different populations, for different species, sorry, for example, for indigo bunting or for spotted towhee, but in general, out of 11 species, 7 of them have significant differences. So in general, I say BRT is actually able to make this distinguish, at least on average. Now, this is not all good news, of course, because you can see also that there is a lot of variation of how probability of occurrence is actually distributed for sinks or source populations. You can see all these outliers for indigo bunting that has really low probability of occurrence values, even though they were estimated to be source populations with CJS-pop. So there's definitely room to grow. There's definitely room for improvement for species distribution models. So in conclusion, I think I can say that species distribution models are poor predictors of species demography. Much spatial variation is left unexplained, and this is the most important part for me, because just building species distribution models and immediately assuming that they directly represent abundance or they directly represent intrinsic growth rate, I think is not safe. So I don't think this is an easy assumption that we should make when we are working with species distribution models. And why is that? It could be because of the differences in processes of occurrence and demography. Occurrence, in the end, a quite high-level thing. A lot happens until you get to occurrence, from demography to occurrence. So that could be a mismatch. So just having a certain demographic parameter may not capture all the processes that gives us occurrence, including our sampling processes. So this could be one of the reasons why there is such a mismatch between species distribution models and demographic parameters. There could be, however, another difference in terms of processes. There could be difference in temporal versus spatial processes. Now, in demographic models, especially for macrocapture model, the environmental signal that we're modeling to fit data to survival and fecundity is coming from temporal data. It's coming from temporal variation. But for species distribution models, it's usually you average across a certain temporal time period here. So when you model that using species distribution models, that kind of data, what you're modeling is actually cell-to-cell variation, population to population variation. And there could be big differences in temporal and spatial processes, especially for the relatively short time periods that we are usually working with. For example, here I'm working with only 17 years. So if there's that kind of a mismatch between temporal and spatial processes, that can also lead to a mismatch between species distribution models and species demography. And it can even potentially lead to that negative relationship that we see for some species. I think this is an exciting future research subject, and definitely we need to spend more time to understand how species distribution models are directly related to species demography. There's definitely not enough study out there about this. The good news, however, is that species distribution models can estimate, at least boosted regression tree, higher probability of occurrence for source populations compared to sinks. So there's a threshold effect at intrinsic growth rate equal to 0. And this is what we want to see, because this is the threshold where we determine whether a population is a source or a sink. So at least on average, we can say that higher probability of occurrence is more likely to indicate a population's ability to sustain itself. So rather than try to come up with a direct correlation between species distribution models, species distribution model outputs and species demography, I think this is a more productive and a safer way to actually interpret species distribution models. It simply tells us about the population's ability to sustain itself. And this is especially important for conservation decision-making. This is important information. And I would like to see more studies actually on this to show that if we can generalize these results to, for example, to different species groups. So this was the end for species distribution models. And like I told you in the last chapter, I'm now going to talk a bit about more about temporal patterns rather than spatial patterns. Now, anytime we make population projections under climate change using population models, they are plagued with a lot of different causes of uncertainty. Here I'm showing results from a study conducted on 2017. You can see that after the data, after the period where they have their data, so close to 2010, and once they start projecting the population size of this bird species, you can see that there is this cone shape and there is a lot of variation. There's a lot of uncertainty coming from different sources such as sampling variance, environmental stochasticity, climate stochasticity, and climate scenario. Now there's also this cone shape here because this is actually a density independent model. So the variance here, the uncertainty here is increased quite a lot. Now, disentangling causes of uncertainty in population forecast is rare. This is one of these rare studies. What's even rarer, or rather what I have never seen, is actually how these uncertainties interact with each other, because they do. They are not acting in isolation when we make population projections. And these interactions have not been quantified before. And this is what I'm going to show you in this chapter. In order to do that, I am going to build population models. I'm going to build population models for tufted titmouse. These population models are parameterized. These state-structured population models are parameterized by CJS-pop. Here in this state structure matrix, fecundity is a function of density and weather, and survival, juvenile survival and adult survival is a function of density and weather too. And using this state structure matrix and the climate change data that I have, I can project the populations of dotted titmouse from 2020 to 2080. And I am going to only focus on 3 causes of uncertainty because I would like to have quite a certain focus on certain interactions. So I'm just going to have 3 causes of uncertainty. The first one is the climate projection uncertainty. I'm going to use I used 19 general circulation models. So for each model, I ran a population model, basically. For each GCM, I ran a separate population model. I did a separate projection. So that led to 19 replicates. I then did temporal environmental uncertainty. This was basically, this is environmental stochasticity. And to see the uncertainty effect of environmental stochasticity, I built 100 different population projections, 100 different replicates. And for parameter estimation uncertainty, I did random draws from the posterior distributions of the parameters that was estimated by CJS-pop. And for each draw, I ran a separate population projection. So here I did 1,000 replicates. My posterior lengths are 10,000. So I use 1,000 replicates to generally show, generally represent a full posterior distribution. And I did a 4th kind of uncertainty. This is not a different kind of uncertainty, but rather this is the combined uncertainty. So in this combined uncertainty projections, for each general circulation model, I did 1,000 replicates, random draws from the posterior. And for each random draw from the posterior, I did 10 replicates of temporal environmental uncertainty, so environmental stochasticity. So for combined uncertainty, I had 190,000 population projections to look at how the population projections varies among the replicates. So the first thing I'm going to do is to look at the variation in total abundance. So I do these projections, each population separately, and then I add up these abundances, and I look at the variation in total abundance across all replications from 2020 to 2080. But then I'm going to specifically focus on, combined uncertainty, and I'm going to do various partitioning on the combined uncertainty on a 3-way, using a 3-way ANOVA structure, so that I can look at how these causes of uncertainty actually interact. All right, so here's the first bit of results. On the y-axis, you can look at, you can see the population size, and the x-axis, we have years from 2020 to 2080. For climate projection uncertainty here, each colored trajectory is a separate general circulation model. The black line is the median population trajectory. And in all of them, the gray line is the 95% credible interval of these projections. And in these 3 cases, you can see these white trajectories, and they are just individual replicates for these different uncertainties that I applied when doing these projections. So one thing you can notice that if just on their own, without looking at combined uncertainty first, if you compare climate projection uncertainty, environmental uncertainty, and estimation uncertainty, we can see that the variability, the uncertainty caused by climate projection and parameter estimation is very, very similar. It's very close. Excuse me. And for environmental uncertainty, it is quite low compared to these 2. However, when we combine all of them, the uncertainty increases quite a lot. And this is not just a simple effect of just adding up all of these uncertainty that we estimated when I just applied them in isolation. This is more uncertainty than just adding them up. So this supports my hypothesis that there's definitely some kind of interaction going on between these causes of uncertainty. So this is what I want to do now. Now I want to disentangle this variance that caused by different factors, different uncertainty factors. Now here in this table, I'm showing you the percent variance explained in abundance across all replications. C is for climate projection uncertainty, E is environmental uncertainty, and P is for parameter estimation uncertainty. Here, I want you to first focus on the fact that our environmental stochasticity estimate is zero. It's not, of course, zero. It's just for single significant digits. It rounds to zero. So we can say that the environmental stochasticity is quite negligible, actually. However, if you move to if you look at the interaction between environmental stochasticity and parameter estimation uncertainty, we can see that much of the variance here in years 2050 and 2080 is actually explained by that interaction. So here, when it's acting in isolation, when it's just we're using the point estimate, it causes a negligible effect on the uncertainty of population projections. But when it interacts with parameter estimation uncertainty, suddenly it just hits 60% of the variation explained. So that's a quite important result, I believe. And I'd like to have a bit of a deep dive into what I mean when I say the interaction between environmental stochasticity and parameter estimation uncertainty. So here is a basic model, a linear model, the way I do in CJS-pop for survival. A is for density and beta is the slope for density. There's beta 2 for slopes for our the variables, the environmental variables that I use in the model. This is just a hypothetical case, so I'm using only 2 environmental variables here. And this epsilon here is the time-dependent random effect. And this is what environmental stochasticity is in this model. This effect is coming from a normal distribution, and sigma squared S gives us the standard deviation of this normal distribution, that standard normal distribution that's centered on 0. So this whole thing here is what gives us our environmental uncertainty when we are modeling this population projection. In each time step, there's a separate random effect here for a given population that is coming from this distribution. Here's the thing though, I also estimate sigma squared S. So this is not only about here environmental uncertainty. Sigma squared S need to be estimated from the data. So even if I have a point estimate for sigma squared S, yes, I can do environmental uncertainty. However, there is also uncertainty on sigma squared S itself, that is the parameter estimation uncertainty. And this is the kind of interaction that I'm talking about when I say there's interaction between parameter estimation uncertainty and environmental uncertainty. Because if I use the point estimate for sigma squared S, yes, it can be negligible compared to others. However, if I use the full posterior distribution of sigma squared S, there are such values that make sigma squared S so high or so low that when I do in some iterations, these projections with those really low or really high values, that we end up with this really high uncertainty in our total population projections. So this isn't only about using the point estimate here, but this is about making use of the whole posterior distribution and how that whole posterior distribution of environmental uncertainty affects our population projections. It is great that we have a really low point estimate. However, it is also important to be able to incorporate these whole posterior distributions to our projections, because they can end up being the major source of uncertainty. So what do we do to reduce this environmental uncertainty in environmental stochasticity? Well, more data helps, as always. More years, more populations. And this is good news because I used only data from 1992 to 2008, and there's already 12 years of data that I did not have access to. So already we might have the option to just reduce this environmental stochasticity. We can also change the model structure. Here, I did only a temporal effect. I assumed that this temporal effect was the same across all populations. A more realistic version would be, for example, a spatial autocorrelation in these temporal effects. So populations closer to each other have similar random effects, and populations far apart can have different random effects. Now, this might also help reduce uncertainty. For example, when I add density dependence, it stabilized my population projections a bit. So it had less uncertainty than the first study that I showed you. Here's the issue though with model structure, and there are a lot of things that we can do for changing model structure. In general, when we do things like adding density dependence or spatial autocorrelation, We are not immediately 100% sure that this is going to reduce our uncertainty in population projections, because each added parameter also comes with their own uncertainty. So yes, you might reduce uncertainty in one parameter, but then you might have increased uncertainty, or rather a new uncertainty in a parameter that was not in your model before. And that brings about the question if there is a fundamental uncertainty that we can never reduce, whether this is environmental stochasticity, whether we call this environmental stochasticity, or we call this parameter estimation uncertainty. And I believe going into the future, especially when we are turning, we are making ecology a bit more forecasting based, I think we are going to have a lot more discussions about how to incorporate uncertainty, how to estimate uncertainty, how to incorporate it into our decision-making process. So in conclusion, I would like to wrap everything up. Small-scale processes, survival, reproduction, density dependence, can explain large-scale macroecological patterns. This is what I showed with the vital rates mechanism. So this is not a futile experience. This is not a futile attempt. I think it also makes it more useful if we have these small-scale processes to actually associate with emergent patterns such as species distributions, because then these models, the output of these models become more meaningful when connected to small-scale processes. This was the case when I told you about how intrinsic growth rate, how species distribution models can actually distinguish between source and sink populations. However, there are limits for bottom-up approaches using only small-scale processes because of the compounded uncertainty that I was talking about. If you just start modeling uncertainty from the population level, they are going to add up when we look at the total abundance across all of the distribution of this species. So again, just repeating myself here, that it is extremely important that we talk more about uncertainty and we do more models about uncertainty and how we incorporate uncertainty into our population projections. Now, this was this was a very long presentation, so I'm going to be super quick to just do the acknowledgements. I would like to thank my committee. They molded me into the scientist that I am today, so I'm very grateful to have such a committee. I would like to thank Melissa, Marta, Donna, and Fumio. They always have students' backs, and as an international student, it is great to have them, because you just feel safe when you know that they have your back. And none of the photos that I showed was my own, so they are all coming from Wikimedia. I can send you the links if you want to. I would like to thank our lab. They supported me all these 6.5 years, so they're the best lab one can have. I'd like to also thank our former postdoc, Kevin Shoemaker. He was a great mentor, and he affected all of this, every work that I did, as much as any committee member. I'd like to thank all my friends. They, again, just supported me along the way. We had fun. I was very lucky to be part of this this community. You see here, I feel really lucky to be part of this community. I'd like to thank my parents. They both have PhDs, so I'm just following in their footsteps. So I feel incredibly proud to be able to do that today. And last, of course, I would like to thank my wife. We started this journey together in college. And it's great to be just doing this journey with her together. And she's actually at a separate presentation right now, so she's not here with us. So literally, that's how we do things in parallel. And none of this would be meaningful, I think, if we didn't all of this together. So thank you all for being here. I'm sorry for such a long presentation. I will be very happy to answer your questions. And Anna will unmute you if you have any questions.
And you can also ask questions in the chat, and then I can read them off if your mic's not working or whatever it might be. Okay, so let's see here. Okay, Alyssa, I'm going to unmute you first. And so you should be good.
Sorry, that was just me applauding a great.
Oh. Okay. Sorry. Okay, so I will mute you. I'm assuming, okay, I see Michael. I'm going to see if I'm going to unmute him.
I will also applaud. Great job,
Thank you.
Bilgecan. So my question is sort of relating the relationship between density dependence and then survival and fecundity that you spoke about. Most of the songbirds, like the ones that you were looking at, have relatively low survival and high fecundity, at least compared to like the seabirds that we study or raptors, other large birds. And I'm wondering if that might affect why you saw a stronger relationship between those parameters and the the population, so the abundance or the intrinsic growth rate, and whether or not you might expect that those, the stronger relationship with survival and weaker for fecundity to change if you were looking at, say, other species or, for example, insects, which would have, an even stronger skew towards low survival, high fecundity.
So thank you for that question. So there are a couple things there. The first one is that I don't really know in terms of like actual density dependent strength, because one of them is on logit scale and the other one is log scale, and the values are, one of them is just limited between 0 and 1, and the other one can be anything depending on the species, like let's say 0 and 10 or more. It's hard to say which one is actually stronger. That's not really an easy comparison, I think, to make. Maybe in terms of how much proportion that they decrease, like half an increase in survival versus half an increase in fecundity. But that comparison is hard. Also, actually, my expectation, to be honest with you, would be to have lower density dependent strength on survival, just for the fact that they tend to have lower survival. Because if you have really high density dependent strength on low survival, for example, then it is going to affect quite dramatically if you have a high density dependent strength. Like, it's going to decrease that survival values to the point that it might be very hard for population to exist. So this is not exactly actually my expectation from what you told me. But you're right. Like, these survival values that are estimated are not super high. They tend to be around, like, at most like 0.5 or 0.6. Like, we don't see survival values such as 0.8 or 0.9, right? But also, I don't know exactly how that, depending on how high that density dependent strength is or how low it is, that would directly translate into a pattern between the density dependent strength and the abundance or intrinsic growth rate. So I don't actually have an expectation for that. And CJS-pop did not really force anything to give you that kind of a pattern back. Now, it makes sense mathematically that if you really have high density dependence, you can have a bit lower density dependent strength and maybe in an opposite direction in fecundity, just because in the end, these need to stabilize around a carrying capacity, around the population equilibrium. So if both have really strong density dependence, you may not see that kind of equilibrium. So there might be a point where you have that kind of pattern, that opposite pattern. However, it didn't have to be turned out that way. Both of them could have been weak, or you could have seen like exact opposite patterns of what I saw, like higher survival for higher abundance and lower fecundity for higher abundance. But that was what I got. And right now, I don't unfortunately have insights into what is going on there, at least for this group of species.
All right. So, Michael, do you have any? I don't want to put you on the spot, but I'm assuming I'm going to move on unless you say otherwise. So, the next question is from Ellie, and it is in the chat.
I have actually read it, so I can immediately answer that.
Okay, sounds good.
So here's the issue with trying to explain these things with individual life histories. Now, this is quite variable that are There are migratory birds, resident birds here, different nesting habits, different, some of them are omnivores, some of them insectivores, some of them feeds on plants. However, 17 species is not large enough that when I separate them into these different life history groups, that I actually start seeing patterns here. So I need to have like 40 or 50 species maybe to be able to assign species to specific life history groups, and then to be able to say, oh, this is the pattern, this is the pattern that I see, and this is how life history affects, you know, abundance or intrinsic growth rate, or the relationship between abundance and intrinsic growth rate. So that's not something, unfortunately, I can do with the data that I have. Some groups, some life history groups just end up with like 2 or 3 species, right? So I cannot generalize from those patterns. And I don't really like to speculate after the fact, just looking at these you know, species and say, oh, this has this kind of life history, so that's why it must have intrinsic growth rate, because, you know, that's just ad hoc speculation and I don't really have the data to show for that. But this is a great point you're making, Ali, because this is exactly what I want to do in the future. Not limit this to only 17 species, do this for more species, so that I am not only showing you that these patterns exist, but I can actually explain why these patterns exist depending the ecology of the species that we are working.
Okay, awesome. So the next question is from Doug. So Doug, I am going to ask to unmute you. So you should get a notification that lets you unmute yourself.
Okay. Firstly, Bilgecan, that was absolutely a terrific talk. I mean, outrageously good.
Thank you.
Of course, I understood only the biological end more than the details of the statistical end of it. But I had 2 questions, which is, one, have you had any opportunity to talk about your work with Bob Holt, who is a very friendly person? He would love to know what you've done. And then secondly is just following up, it's almost the obverse of the previous question, which is, to what extent do you feel as if the set of species to analyze has to have some degree of homogeneity? Because as you pointed out, you've got, you know, a number of different sort of ecologically very distinct kinds of species there. But, you know, you could imagine throwing in herons and ducks, you know, but presumably you wouldn't want to do that because their biology is so very different. And I'm curious then about, in some sense, are there any rules for what would constitute a set of reasonably comparable species?
So, okay, the first one. No, I did not contact Bob Holt, and that is, you know, my Achilles heel. I definitely, I will. These are not so published yet. I'm hoping that I will be able to present this in other places, and along the lines of publication, I will be able to talk to him. I hope that that's going to happen. And for your second question, yes, that's a really great point, and it's absolutely affects how you look at abundance-occupancy relationship. I mean, you can think of like the extremes of it, like if you add mammals to that, like how would you expect, you know, to see abundance occupancy in such variable life histories for all of the species. There are no rules, exact rules established. There are extremes in literature, like you can only use the same genus kind of extremes. Now, if you only use the same genus, yes, you might see maybe a positive abundance occupancy relationship, but I think it becomes less and less interesting as you move down further line. Like if you are just coming up with rules to say, this patterns happens only if I have this and that and that and that, and then it's not really a, you know, a general pattern at that point. So that is why I don't favor doing that, like going really to, for example, genus level. However, this is great in the sense that it should be quantified. It should be, it should be quantified not in terms of, I think, species taxonomy, because in the end, species taxonomy is all about the life history here. even like when I say things like, is it similar density dependence? Is it similar response to the environment? That you're actually bypassing those processes when you just focus it on just the same taxonomy of the species. So it's better to, in a way, like liberate this from just the taxonomical way of looking at this and try to explain how this actually comes about, which processes are involved when this comes about, when you have different species coming from very different life histories. Of course, the goal here would be to, this is going to be arbitrary at first. You know, you can go with family, you can go with order, you can make these different groupings, or you can decide on different groupings of species for raptors, for example, even within raptors, depending on like you can do separately for vultures. And then step by step, you can group those species into, depending on specific hypotheses, depending on their specific demographic estimates, you can group them and see how the abundance occupancy changes. And depending on it becoming more weak or more positive, that is going to give you the actual process that leads to those patterns. Of course, the limit here is that we just don't have the means to do it for that many species. That's the thing. Like, there are more than 300 species captured in the MAPS data sets, and only 62 of them I was able to apply CJS-pop to, because they just don't have juvenile recaptures. And out of 62, I was able to apply CJS-pop actually to 42, because 20 of them have really large data sets. And yes, there's such problem as very big data set, because your model then can take a month to run. And you know, you want to finish your dissertation, so you're not going to run those models like that. So out of the 42, 8 did not convert, so my models did not give any results at all. And 17 had positive density dependence, which did not have any ecological meaning at all. So that was like my model selection method. So I ended up with this 17 species that I was going to test this relationship with, right? So I did not really have the luxury to have those groupings. It was just, how do I do this with many species that gives me results? Like, that was the approach. But going forward, I'm optimistic as we develop these models more and more, we are going to be getting more results from more species. It's just that I really want people to focus a bit more on processes as much as they can.
Great, thank you.
You're welcome.
Okay, so the next question, and thank you for that great question, Doug. I'm going to remute you just for the sake of the process. Liliana has the next question. So Liliana, you should get a notification to unmute yourself.
Yes, I am unmuted. Can people hear me? Yes.
Yes.
Yes, we can.
So I have a question that's more kind of like beyond the results that you presented. I think some of the things that you showed make a pretty compelling case that the way that people have been thinking about these distributional models it does not really track, right, like the reality of the data when you're looking at the population. So what would you, if you had to kind of summarize this, how would you communicate to people who are saying, well, this is the best that we can do for projecting into the future, climate change is speeding up, we need to do something? What would you say to them? What would you recommend that people use who are in that situation?
So I think in that, what I said, it's about a population's ability to sustain itself. Now, it is a very simple explanation, but that is how I am going to try to think about here species distribution models. It's just that if you have your species distribution model giving you a higher probability of occurrence or higher suitability, it just means that in the future, let's say under climate change, you predicted higher suitability for a given population, it means that that population probably is going to be okay. But that's the extent of what you can say. It is going to be able to sustain itself. If a given population or if a given cell has really low occurrence or suitability, the only things that you can say, if it is currently occupied, there is a good chance that it can go extinct because it won't be able to sustain itself. So for it not to go extinct, it is going to need support from outside populations. So one thing that you can look at, are there close by populations that have higher probability of occurrence? If you don't have that, then that means, you know, if you leave it as is, it's just not going to be, you know, there in the future. So you might need some, you might think about some artificial, you know, relocation studies of some species, maybe relocation applications of some for some species. If it is not already occupied, and if it is a low occurrence, then you can say that even if it gets dispersal in the future, it won't be able to probably sustain itself. So it's again going to be all about dispersal. Is it going to have, for that place to be occupied, it's not only about whether the species is going to disperse to that individual cell there in the future under climate change, but it's also about, is it going to be able to disperse around that cell where species can actually establish so that it can feed into that cell with the low occurrence. So this is I had a bit of a shift in thinking in species distribution models to more source sink dynamics rather than looking at it as we have really high abundance, and with species distribution models, if there's high probability of occurrence, there's high abundance. If there is high intrinsic growth rate, there's high probability of occurrence. So that has a bit shifted in my thinking, I believe. So those would be my general suggestions. However, also, I think we need to be doing more studies like this, and quickly. There is I think in a review said 6,000 or so SDM studies published in the last 20 years, and 3,000 or something of them are on biodiversity assessments. But the meta-analysis that I showed you was only on 30 or 40 studies. So we are not doing enough to establish the theoretical basis of species distribution models with data, and we are immediately jumping to conclusions about how we can use species distribution models. I think that is also something that I really want to emphasize going into the future.
Fantastic, thank you.
No.
Okay, thank you, Liliana. Next, we have Raphael, so I'm going to ask to unmute.
Hi, can you hear me?
Yes.
Yes.
Hi, Bilgecan. Thanks for the talk. It was great. I also like how you explain your methods and your models and your statistics, and very intelligible. So my question is also about the same thing that Liliana just asked, which is the ability of the SDMs to predict whether it's a source or sink population, or predict the abundance or the actual valued intrinsic growth rate. So unless I misunderstood, when you try to predict the abundance or the intrinsic growth rate, you're trying to predict a number, a quantitative value, a real value. When you're trying to predict whether it's a sink or source, you're just trying to predict a binary, right? Is the R greater than 0?
Exactly.
So there's a way you can say it's a lot easier to predict whether something is greater or below, above or below a certain threshold than what that value actually is. So I guess my question is, was it expected that the models would do a better job of predicting source versus sink than actually predicting abundance or R? Or do you think your result, it goes beyond this, predictable methodological issue of you trying to predict a lower information variable, and it has something to do with what's going on with the populations, and you mentioned dispersal as one possibility?
Great question. So it was not expected for two reasons. First, we are usually taught that intrinsic growth rate is not something that we can get at with species distribution models, because it's more about fundamental niche, and what we are getting at is actually the realized niche. And with the realized niche, you have these two complicating factors, the dispersal and the biotic interactions. So dispersal and biotic interactions do make things really complicated. Especially, I think they make them complicated when we try to predict them as is, like the abundance and intrinsic growth rate, like you said. But the general idea was that they won't necessarily be able to distinguish here what is a sink and what is a source using species distribution models, also because of those reasons. Because your data simply might have if it is dominated your occupancy with a lot of sourcing dynamics, if you have a lot of sinks in your population, that basically gives you the signal of as being a presence, then also if you have biotic interactions on top of that, that you have absences that you would normally have, for example, presences, then the things get quite complicated real fast. So you don't actually expect to see that kind of ability. At least I didn't expect to see that kind of ability from species distribution models to make that binary distinction. The fact that we can see, I think it's good news. It's good news also because the distribution models that I built are very generic. I think like we can definitely do better jobs in terms of building these distribution models. However, I wanted to build these generic models because I wanted to establish a baseline here. Like it's not about just, not every species has a lot of data. Not every species we have a lot of data other than what the environmental variables are and what the occurrences are. So I wanted to look at what we can achieve with just the occurrence data, just with the environmental variables, and depending on how we generate our pseudo absences. So there is definitely room for improvement for species distribution models there, and they might, with the certain improvements, they might end up becoming better predictors for demography as well, and also for distinguishing sources and sinks. But I think in general, that part is good news, unexpected good news for me, that they are able to distinguish between sources and sinks. I don't know if that answered your question.
Oh, it does. Thank you.
Okay.
So I don't see any more questions in the chat, and actually it's been exactly 20 minutes, which was how much time you wanted to allocate to questions. So I think with that, we can call it a wrap with questions. I'm not sure if Stephen says anything to wrap this up.
We are going to, I think, meet in another room. Thank you everyone for being here. Thank you for your support. I was really happy, really excited to share all my results with you. I hope you're all doing well in the middle of the pandemic. My best wishes, best of luck. And Stephen, if you're not going to say anything, anybody, I am going to end the meeting.
Okay.
Thank you, everybody.
All right. And
I'm super happy to see you today.
Let me say one more thing. Today around 5, we're going to have Friday Social is going to be celebrating Bilgecan. And so I'll send out an email later today with that information. So just be on the lookout for that.
See you all today at 5.